Thank you everyone for having me. It's really lovely and bizarre to be in front of people again, talking. I hope my talk is interesting. I've been told that mine and Alex's talks are very different. Mine's very much based on the research that I'm doing at the moment and results from that. So it is quite exciting for me at least, and I hope that shows. So my name's Jonathan and I work in a research group at the RILD, which is an institute at the Wonford hospital site. It's part of the university, and my group specialises in epigenetics, particularly in the field of psychiatric disorders, and my work specialises in fetal brain development. So I will warn you that there are references to miscarriage and abortion and infant mortality. That's simply because that's where we get our tissue from, unfortunately, and I'm happy to take questions about that at the end as well if you were interested or had concerns. But my talk is about gene regulation of neurons in the developing brain. So I'm going to take you on a little journey and teach you about what it is that we do research into in our group. So first off, what is epigenetics? So I hope most of you should have heard of DNA, but every single cell in your body will have a nucleus in that contains genetic material, which is your DNA, and that codes for all the proteins in your body that make you who you are. But every single cell in your body will contain exactly the same DNA, but they have different functions, different phenotypes, different looks to them, and that's because it's how the genes are being regulated and controlled, how these different genes are being expressed in these different cells that creates this different look, and not just at cell levels, but obviously overall body levels as well. And this was characterized by Conrad Waddington, who created this concept of the epigenetic landscape. So you start with one cell in very early development, that contains all the genetic information for every single possible cell in the human body. And as these cells start multiplying and differentiating down different lineages, like the trouser legs of time, you're starting to develop these different regulatory mechanisms that force a cell to become more neuronal-like, more nerve-like, or more like a skin cell, or more like a blood cell. And so that's one type of epigenetics. And that determines cell type, but it can also determine your health and your well-being, and is influenced by the environment. So some of them are transient, some of them are permanent for your lifetime, and they will change over time as well. So the type of epigenetic mechanism that my group studies is DNA methylation. So you have your DNA structure, and your DNA is made up of 4 bases, represented here by the C's, G's, A's, and T's. DNA methylation only happens on the cytosine residues, so the C residues, and that happens with a transfer of a methyl group onto the cytosine, creating methyl cytosine. And this is really important because this is a general principle, and obviously in biology there's always exceptions to the rule, but generally, if a site on a gene is highly methylated, it means that the proteins that promote gene expression cannot bind to the DNA, and therefore those genes cannot be expressed. If you remove that methylation, the DNA is able to open up like a zip, and these proteins can attach and start to express the genes, and those are unmethylated sites. So we discuss this issue as if it's like a regulatory mechanism of turning on or off a light bulb. There are other really good analogies to think of it. So you can think of a piece of music where the notes are the DNA and the instructions like crescendo and diminuendo are the epigenetic mechanisms controlling how you're playing the piece of music, or a piece of playwriting, yes, whatever it's called, script, where the text is written down, but the stage instructions are telling you how the piece is being performed. So we can look at this from opposite angles. So we only look at the DNA methylation in this talk today, but we have So we have the DNA at this end, the helix, and that's really tightly coiled, wrapped around proteins. The proteins are wrapped around more proteins and tightly coiled and compacted to form the chromosomes that you know and love today, and they are found in the nucleus of the cell at this end. And all of these mechanisms are ways for the genes to be regulated, because if it's really tightly coiled chromatin, again, the genes cannot be accessed for expression. But today I'm just going to focus on the methylation. And gene regulation is important in development and disease. There are many papers that have looked particularly at twin studies. So in monozygotic or identical twins, you can see in early stages of their life, their DNA methylation levels are similar. comparable, and over their lifetime, they will have different environmental influences, whether they smoked, whether they drank alcohol, whether they had serious injuries. And we expect to see at the age of 50 that we have very huge variation in methylation profiles at specific gene sites of interest. And this becomes really relevant when we look at diseases as well. So particularly in my group, we look at psychiatric disorders, and my focus is on autism and schizophrenia. So if you look at autism, in monozygotic twins and identical twins, less than 70% of twins where one individual is diagnosed with autism is the other twin also diagnosed with autism. So we have more than 30% of twins who aren't concordant that we can't understand why, if they have the same genetics, why do they not have the same disease? So there is this environmental impact and particularly gene regulation that we think is contributing to this development of these psychiatric disorders. I'll briefly talk about autism spectrum disorder. So it is a neurodevelopmental disorder. We still don't know what causes it. 60% of cases of autism have an unknown etiology. We have found genes that are associated or may enhance your risk of developing them, but some people might have these genes that associate with the risk and do not express any indication of autism spectrum disorder. It's important to note that terminology around ASD is changing, and people start to refer to neurodiversity and neurodivergence in individuals. And I think that is really important to be aware of that change, but it's also really important to remember that there are many individuals who ASD is still a severe disability, that I have worked with individuals who have regressed in their childhood and are non-verbal, non-functioning, they can't walk, they can't talk, and it's a huge spectrum of disorders, so we have to be encompassing and make sure that we are accepting of those people who are neurodiverse, but aware that for some people it still is a severe disability that we do need to do research into. So my group has published many papers looking at DNA methylation, particularly in brain development, and looking at trajectories across human brain. And we've started to recapitulate this with a larger cohort. So we get access to brain bank tissue banks where we get sent small bits of human brain tissue, post-mortem tissue, that we can extract the DNA and analyse the methylation profiles of these samples. When we look at a whole piece of brain tissue, what we're able to identify is trajectories of methylation changes across the time span of development. So each of these little black dots here represent one individual that we have profiled. This CG site, all you need to know that this is a site associated with this gene OPA1. So some genes might have many sites associated to them in the arrays that we use. But what this is showing is that in the very early stages of development, so we have samples as young as 6 weeks post-conception, we have very low methylation at this site, and across development up to postnatal, post-birth, sadly we have some early postnatal death samples. We see really high levels of methylation at this site. So we're showing here the 3 highest trajectories of increases in methylation, and what this means is that over time, higher methylation means that this gene is going to be less expressed at this time point because it's high methylation means less accessibility. In contrast, these genes, the further you go through development, the less methylation they are, potentially the more expression we see. So AMBER1, I think I remember, is involved in on nerve cells. You get these processes called dendrites, and it's involved in dendrite formation, which is how the cells communicate with each other, which is logical because you want these cells, as development happens, to start being able to communicate better with each other over time. We don't just see these really nice linear models, we also find non-linear ones, which are a bit harder to analyse because we need to start understanding how genes are more transient through development. And not only that, as I said, this is just in whole tissue, and it doesn't actually describe to us what the differences are in nerve cells compared to immune cells or blood cells or epithelial cells in the brain. So we've started to develop a technique. Oh, forget that. I've missed a slide out. We can also add other variables in to pull out other trajectories. So we've actually added adult samples here as well, and we can project this into the adulthood. And we can see, for example, at this end, that we have a gene that in the fetal samples, the male and female samples have the same kind of pattern, and as we move into adulthood, you see a difference in the male and female pattern. So we can see a dichotomy in gene regulation patterns as well. And these are actually autism risk genes. So these aren't identified in autistic individuals. These are just genes that we know could be associated with autism. So as I was saying before, I forgot a slide, the brain has many different complex cells in. I put these images in because I thought they looked really cool and rainbow coloured. These are actually slides from a genetically modified mouse model called a rainbow mouse. So every single cell in the brain, when exposed to a drug, normally tamoxifen, will start to express different coloured proteins, which are identifiable by microscopy. This is the hippocampus region of a rainbow mouse, showing the complexity of the brain structure and how difficult it would be to isolate a whole cell from the brain. So what we do instead of trying to get everything, we just try and collect the nucleus, because that's got the DNA that we are interested in and we don't care about the rest. So Steph in our group has optimized this technique where we take a small bit of brain tissue, either from our adult cohort or our fetal brain cohort, and we homogenize the sample. We spin it through a very dense sucrose cushion, which means that all the rest of the cell debris sits on top, and the nucleus, which is incredibly dense, will pellet at the bottom of the tube. And we can isolate that, stain it for antibodies against the different cell types that we're looking for, and we can use this cell sorting machine to pull out the different cell populations. On our big project, we actually try and collect lots and lots and lots of fractions for lots of different downstream purposes. So we'll look at RNA expression, which is transcription. We'll look at chromatin modification. We'll do different isoforms as well. But today, I'm just focusing on the DNA methylation. And in this cell sorting machine, it's really cool physics and fluid dynamics, which I don't understand. But it's really exciting, and I love that someone has engineered this machine, and it's incredible to see it in action. You get this, what looks like a solid stream of liquid, but it's actually like millions of particles of water, and each tiny droplet should contain one cell that you've passed through the machine. And a really high-powered laser beam will shine at each single droplet and will detect the size and complexity of this object that's passing through the machine, but also the color that is tagged onto it. And instantaneously, it will apply a tiny static charge, which will sort these cells into different tubes for us to collect at the end. Just ingenious. I don't know how it works, but it works, and I use it every day. And in our adult samples, it works really well. Again, every single dot is a cell that's passed through the machine, and we can create these gates that tell us which are the cells, which are stained blue, so we know that they're actually intact cells and they're not two cells stuck together or just debris floating around. And then these are the different markers that we're staining for our different cell populations. And up here you can see that they group really nicely into 3 separate populations, and we can say that these ones are our neurons, these ones are our oligodendrocytes or immune cells from the brain, and these are none of the above. Which is still interesting because they contain rare populations that are really hard to get hold of. And that's wonderful, and we do this for our adult population, but as I said, I'm interested in brain development. And when we tried it on our fetal samples, it just didn't work. So we couldn't see the emergence of any population until postnatal ages. So we had to find, and this was really frustrating for us because NUN is the marker that we used, and it's really well established in the literature, and there's papers even published that say that they use it in fetal tissue, but we can prove that it doesn't work. So we're going to tip the apple cart, which people aren't going to be happy about, because it means that they've lied. Or at least not been 100% truthful, because when you collect cells on this, we can see in adult samples that they group really nicely based on their cell population, and this is lovely. This is like a hierarchical clustering of their methylation profiles. Whilst in fetal tissue, it looks a mess. So we're not pulling out any individual cell population. So we needed to fix this. And we went off and we found a different marker. I skipped a whole story, but it's too long to tell all of it. So we found this other one called SAPB2, which is a known marker, but it just hasn't been used that much before. And there's a few groups that have started to publish papers on this, so it's clearly the new trend. And you can see the grey doesn't come up very nicely, but there is a nice clear cluster here that's a separate population. And we confirmed it in the adults. So here we can see the NUN marker and the SAPB marker, they cluster nicely, so it's the same population in adults, which is nice. And in our fetal samples, you can see that we're putting out a nice clean population of neuronal-like cells using that marker. And that means that we can go on and start to investigate the nerve cells and the non-nerve cells in our fetal samples as well as the adults, which is really, really novel work and just hasn't been done, which is really weird to be at that forefront of science. Like you think when you're a kid you're never going to be there, but suddenly you're doing something no one else has done before, and it's really weird and novel. But in that, we can start to look at that methylation difference in different cell populations, and it's really exciting because we can then compare these neuronal and non-neuronal patterns to the adults, and we can see that this gene, PATCH1, becomes equivalent in adults, or this one, ANX-A4, you get a reversal of the pattern between neurons and non-neurons, and at the moment, we don't know what that means. We don't know the implications of what that means for the development, but that's what we're moving on to do. So this is the state that we're at the moment, trying to create this trajectory for all these different genes in all these different cell populations, and trying to create this projection throughout development, and potentially we can start to look at cohorts of individuals who do actually have psychiatric disorders or neurodevelopmental disorders like autism or schizophrenia, and see whether there are points at development that we see a divergence away from what would be seen as typical of these developmental trajectories, and potentially find a reason why there is neurodivergence or changes in phenotype. So that's my research aspect. So I do need to thank Alice, who's my bioinformatician, who's done so much work on this project. So she deserves all my thanks, and I thank her every day. This is my research group. If you were interested, please drop me an email, or message on Twitter, I don't mind. I'm always happy to talk about my work, as you can see. But I didn't want to stop there. I also wanted to talk very briefly. I don't know how I'm doing for time. Miles away, okay. Yeah, it's fine. Perfect. This isn't my background. I found it online, and I've got the link on the next slide. But some guy created really cool backdrops for Zoom, so if you wanted, go and find them because they're really cool. He does different color coordinations. But I also wanted to talk about the ethics involved in genetics research, particularly around the area of sexuality and gender identity. So although I do study psychiatric disorders and neurodevelopmental disorders, and I'm certainly not equilibrating sexuality and gender identity with neurodevelopmental disorders or psychiatric disorders, but it's obviously clearly a passion of mine, and I want to represent that in my work. But there's clear implications for studying genetics and epigenetics of sexuality and gender identity. And papers have been coming out since the 80s and 90s talking about whether we should be doing it or not. And every year, we went out for dinner before this meeting, and Alex pointed out every year someone does a paper that they get a huge backlash because then we're like, why have you done this? Like, it's not benefiting anyone, you're endangering people. But early on, the discussion wasn't necessarily about that, which it obviously should be now, but back then, it was whether it would benefit society, whether we would learn anything new, because people genuinely thought that sexuality and gender identity were personal choices, and they didn't think that we'd be able to find anything different about our community because they thought we were just choosing this lifestyle. And that's a huge sweeping statement, and I know it isn't true of everyone, but it was a common preconception. But even 2019, this was a project where they did DNA testing, and obviously potentially you're running the risk of finding a marker or a gay gene, like the fat gene, that you can test someone and be like, yeah, you're going to be gay. But that's great for scientists, and obviously I would love to find out why and how, but particularly the media and the general public don't understand the nuances involved in genetics and gene regulation, and they don't understand the term associated risk either. So just because you have a gene doesn't mean you're going to be fat or be gay or whatever. And some genes, some diseases or disorders, you might need hundreds of genes to be associated with it for you to even be partially at risk. But that doesn't come across well in our media today. But there have been some really interesting papers that are published, and they are important to look at because for some members of our communities, they could benefit, particularly, I don't think I linked it here, but there is this discussion on why we exist, and there are several theories of whether having homosexual members of a family could increase additional parenting care within a community social upbringing. So I'm one of 4 children, so that I'm redundant effectively, so I wasn't needed to make more children, and I could be around to help raise my siblings. That's the theory behind that kind of hypothesis. Whether that's true or not, we don't know. Or the other idea is that potentially behaviour traits observed in homosexual individuals could be desirable in a breeding partner and therefore are retained in heterosexual relationships, which is a theory that's proposed in other kind of phenotypes that are seen in other animals and humans. And again, we are too homogenous as a species to even be able to hypothesize on these issues. But then also particularly looking at gender dysphoria. This one was, no, it wasn't that one, this one. This one was fantastic. This was literally a study being like, are people with gender dysphoria happier people after they've gone through therapy? And of course the answer was yes. But it was really good that that study happened because people needed to know that it benefited people. And there was a really, really good Danish study, which I couldn't find the paper for, but I do remember reading. So please, I hope I haven't made it up. There's a really good paper that did functional MRI scanning, which is not just taking pictures of the brain, but looking at brain activity in young teenagers in a Danish population who identified as not the gender that they were assigned at birth. Sorry, I'm talking fast, so do forgive me if I get terminology wrong. And they found that individuals who identified as transgender, their brain functionality associated more strongly with their desired gender than it did with the one that they were born with, which shows that there is a difference in these individuals, and with that kind of help, you could potentially have earlier interventions and better support networks for these individuals who are identified early. At the same time, it raises the same questions about risk, whether you're scanning children and being able to identify them and highlighting them and pulling them out. So it's a really, really delicate situation. And the LGBT Foundation has really good guidance on ethical research on these areas. And I had a really wonderful idea. We do huge population studies, and I would love to create a huge cohort of different sexualities and gender identities. The Kinsey scale was great at the time, but it's obviously found to have its limits, but there's a Klein grid of sexual attraction and behaviour, and it'd be really interesting we know it's not binary, we know that we need to create this whole spectrum of different phenotypes, and then look at their gene regulation in blood tissue or brain tissue, or brain's obviously hard because we have to wait for people to die first, but blood's a lot easier. People are more willing to give you blood, surprisingly. And also in specific cell populations. Risk association here is the term we use in genetic studies. So risk doesn't mean danger, it just means likelihood. So it's not, and again, it's one of the confusing terms that could be picked up by the media. And this guide helps you understand whether the pros of this would be understanding human behaviour and development. I would love that. I would love to know why I'm here or how I came to be. And we could potentially develop early intervention strategies to support particularly transgender individuals or families and support networks. But the cons is that, is the community going to benefit from this? Can we find other ways to deal with this? Can we just be a nicer society anyway? And could the data be used nefariously? And I think that's the biggest issue, particularly when we have countries that are willing to identify people and punish them in whatever way they see fit. And ultimately, because of that last comment, it means that we can't do this work. So that's really sad, but it's the truth of society today, sadly. So that's where the backgrounds came from, so please go and look them up, they're fun. So that was my talk on ethics, and then we were asked to maybe add a little bit about ourselves and our Claire's been looking at later, about ourselves and how we felt about our career in science and stuff. And this isn't a chance for me to offload and you to all take the burden of the weight of my troubles away with you. But I think I'm very lucky. Working in bioscience. Biosciences is very female dominated. In the lower echelons, we're still very male dominated in professorial roles and department leads and stuff like that. But most of my colleagues are women, and I feel like women create a much more diverse and accepting and inclusive atmosphere in science. And I've never particularly felt in my workplace that I need to hide who I am. And I'm very, very fortunate and thankful for that. But it doesn't mean that I've had it easy. So I came out quite young. I was 15 when I realised I was gay, and I came out to my parents when I was 16. And it did impact my life. I was a straight A-star student, and my grades crashed through the floor because my parents weren't particularly supportive or understanding. And I was meant to have aspirations to study medicine, and I couldn't because I got D's and C's in my A-levels. And I had to fight really hard to get back to where I am today. And I'm not a medical doctor, which is good because I realised that as much as I thought I liked people, I don't like people enough. And I'm here. I'm only here because I was dragged here. But I am a doctor. I do research, and I hope to help more people because science helps more people. You know, hopefully we have a broader reach. But sexuality does impact your career, and it gives you a burden that you have to deal with for the rest of your life. And having to process that and accept that is hard for some people. And so it's nice to be able to come to events like this and represent, really. So thank you. Go on.